Good morning, Sam. We'll start at 11. We're gonna wait until maybe 11:01, folks, for a few more people to come in before we get started. And keep in mind too, if the internet does go out for any of you, if you're using the internet on campus, if it does go out because they've been having a lot of problems, don't worry. Don't worry. This is being recorded. Hopefully you all got to see the grades from the exams. I think I've got most, not everyone. I'm still grading 3. I think I've got 3 people left for grading. But that gives everyone a nice boost since this is after the middle of the semester. It does give you a nice boost for the last 2 exams that will be coming up. Any questions before we do get started that you all can think of? Quiz grades should be up for this week too. Thank you all for being so flexible too about doing this today. Of course, Monday's a regular class and we'll have a regular lab. I am also going to let you know when the next test taking session will be, which will probably be in about 2 weeks. But those tests that we did, or those quizzes that we did last week, for those of you who were there, those are already posted under the Bright Space contents. Okay, let's go ahead and get started. Hopefully you all can hear me. And no, you guys are good. I see you all. You all are fine. You don't have to put the camera on. You guys are fine. I know how it feels for some of you, and you have to do stuff. Yeah. So anyway, let's get started today. I do want to continue this chapter. It's a detailed, and it can be difficult, this chapter, because we're also going to get into chapter 24 and then evolution after this. Keep in mind that as we actually go through this, that it's not as difficult when you do the Punnett squares. Okay, I promise you, it's not as difficult when you do the actual Punnett squares. Okay, so keep that in mind. Also, I'm going to be giving you a few Punnett squares to practice next week. that will help you a great deal. Okay, so let's actually, Evan, you should be able to get in. Let's actually go ahead and do this independent assortment, second law of Mendel. So remember, first law of Mendel is the law of segregation, which states that the chromosomes, homologous chromosomes, separate during meiosis. They separate during formation of the gametes. That's critical. Okay, so really, really critical to keep that in mind. As we're talking about this law of independent assortment, now we're talking about two or more genes, two or more pairs of alleles. So a pair of factors is a pair of alleles. Okay, pair of factors is a pair of alleles. And a pair of alleles, you know, is one gene. Okay, so law of assortment refers to more than one pair of alleles assorting or separating independently. Independently simply means that they separate totally in their own way from other genes. They're not at all linked. So the key word is these genes are not linked. Okay, not linked. And I'm going to put this in chat too for you. Not linked at all. All right, so having said that, that means the following, that they're not linked, okay, for everyone. And I'm just doing this to everyone, okay? Not linked means that genes, which are pairs of alleles, are independently assorting. That is independently segregating. Okay, so keep that in mind. All right, so we're going to start a two-trait cross. You already know that a single trait cross, and just so you know, guys, there's a lot I have to go through here, so hold on to the chats. If you do have a question, hold on, because I'll probably answer that near the end, okay? So here, thanks. Two-trait cross basically means that since this fulfills the law of independent assortment, we're going to have two or more genes. If you look at these right here, FFSS. Remember I said at the end of class the other day, you've got two different letters. Those are two different genes. Okay, so I'm writing this in here on the slide. Those are two genes. You know that because they're two different letters, F and S. Okay, so now we're going to do something called a dihybrid cross, not what you see at the bottom of this slide, but hold on. We're going to do a dihybrid cross, which is the following. It is right down here at the bottom of this slide, okay? We're going to see that a dihybrid cross is the following. I'm going to put this into chat for you, for everyone, okay? So here it is, FF, SS, and again, heterozygotes for each gene, cross. So notice heterozygote for the F gene, heterozygote for the S gene. Okay, the little X means two cross. Little X means two cross. All right, so that's key here. This is a true dihybrid because remember, and this is in chat, hybrid means they are heterozygous. Heterozygous for that gene, for that trait. Heterozygous for the F trait, heterozygous for the S trait. Heterozygous for two genes. Ultimately, two traits. Okay, so of course they go through their own example here, freckles and finger length, which is a bit different, but now moving here, there it is, right there. And I want you to note that. This is a perfect example right here. Okay, so heterozygotes for 2 traits. We could also do 3 traits. We could do 40 traits, but we're only going to stick to 2. And the only thing you need to know about this is there is a phenotypic, not genotypic, but phenotypic ratio from all dihybrid crosses, which is what this is. This is a dihybrid cross right here. 2 hybrids for 2 what? For 2 different genes. Okay, phenotypic ratio is 9 to 3 to 3 to 1. Okay, you do not need to know what the 9331 means, but you could probably figure it out. Okay, the first one is going to be dominant, and the last one is recessive. Okay, and then they're kind of reversed in the middle, but they're a mixture in the middle, as you can see. So 9331, I'm not going to be asking you what the 9331 means. So this ratio is always expected for a dihybrid cross when dominance is present, when we say simple dominance here. To actually illustrate this, you can see 9 for here in this freckles and short fingers is considered the dominant phenotypes. Remember, this is a phenotypic ratio here. So we're talking about what? This, 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 and let's say that's 6, 7, 8, 9. There you go. So 9 out of the 16 boxes illustrate a phenotype that is dominant for each trait. Dominant for each trait. That way you can figure the rest out. Of course, the one that's pretty easy, that's right down here, that's way in the corner, that's recessive for both traits, which is no freckles and long fingers. If you're interested, you can look into that some more, but I'm not going to be testing you more or further on that. This particular slide shows you how they got the dihybrid parents in the first place. How did they get the dihybrid parents? Here's the dihybrid right here. It's from a so-called F1 generation. So something new to learn. Generations, because we're going to be adding and talking about something else in these Punnett crosses. You need to know definition of a P generation. P generation simply refers to the the parent generation here. In this case, the parent generation happened to be purely what they call purebred for dominance, because for each trait, they are homozygous dominant. The second parent, whom they cross with, is purebred recessive for each trait. That cross results in offspring, children, who are completely dihybrid. That is heterozygous for each trait here, as you can see. It's nice because in the middle, they illustrate the gametes separated out. Okay, notice there's just one allele, meaning one F and one S in each gamete, whether it's sperm or eggs. And then I do want you to know the F1 generation stands for the following. I'm putting this into chat. Okay, the first filial generation. That is the first offspring from the cross of the parental generation. Okay, so first filial. There's also a second filial. There can be a third, and obviously 400 or 4,000 from that. So dihybrid cross is the actual crossing, and note where I'm drawing this arrow, please. We saw this just back here. Remember, this is the true dihybrid cross because you're crossing two dihybrids. So one big F, little f, with one big F, little f. Of course, they're also big S, little s as well. Dihybrid. Probability, if you want to look into this more, for that 9:3:3:1 ratio, which is phenotypic that we came up with. So memorize that ratio. And remember, what was the ratio? And I have this, just so you know. Oh, hold on a sec. Okay. Justin. Okay, there you go. Hopefully that should. Thank you. So here, I want to remind you that what was the ratio for monohybrid crosses? Phenotypic ratio was, and this is in chat, not 9:3:3:1. What was it? Easier, 2 numbers instead of 4. 3:1. Okay, remember, because we added the genotypic ratio of 1:2:1. Okay, we added the 1 plus 2 to give us the 3, the phenotype, because 1 represented, remember, homozygous dominant versus, and I'm writing this into chat, versus heterozygous, the next number, the second number, and then the last one represents homozygous recessive. Okay, so there you go. It's all there in chat. All right, so moving on. Now let's talk about pedigrees. These pedigrees are different. These are not crosses. Many of you have heard of pedigrees. Pedigrees, definition, are charts that illustrate the history of a family with certain traits. And these are usually genetic traits. Of course, it can be done on animals, even plants, as well as various other living things. You should know that males have traditionally been designated as squares. Okay, females as circles. Okay, I do want you to know that. So in pedigrees, we're also going to see that there's going to be a P generation, an F1 generation, and F2 generation, which is the second filial generation. And you can keep going, as you can imagine. If an individual has a disease, then this is shaded, either the square or the circle. If they do not have the disease, it is kept unshaded. All right, so pedigrees use these different keys. Here's an example. At the very bottom here, we can see, and this is initial. So you can see the parental generation would be the male crossing with the female. Okay, both are affected by that disease because it's shaded. All right, both are yellow. They're shaded because they have the disease. But in the first filial generation, they happen to only have one child so far. They could have more in the future for the same first filial generation. That child is unaffected. It has no disease. It does not have that particular disease. So now let's actually look at this in more detail. And we're going to talk about autosomal recessive and autosomal dominant pedigrees. I don't expect any of you to be able to read these. If you go further in general biology, they do expect you to interpret and guess as to whether a pedigree suggests either recessive or dominant traits. But I will tell you it will be extra credit in terms of exam questions. So number one, this is what we would consider to be the parent generation, first generation, second, and so on. First filial, and so forth. Okay, so parent generation, notice mom, and they do illustrate this. Mom and females always have a cross attached to a circle. The males always have an arrow attached to the circle. They're showing you the mom is affected. Okay, mom is affected. She is homozygous what? What's her genotype? You can put it into chat. Homozygous, not dominant, but recessive, right? Homozygous recessive. But dad, we don't know the complete genotype. Okay, we do not know. We do know dad has at least one dominant allele, one dominant allele. He could have either a second dominant allele or a recessive allele. It's not both, it's one or the other. So from looking at the different generations, generally people can determine, okay, well, was this more a father who was homozygous dominant, which would be this, or is he heterozygous, which would be this? And this is the fun that they do in General Biology 1 and 2, because looking at the first generation, no one's affected. Nothing is shaded yellow. In fact, some of these children, we don't know their complete genotypes, but we do know because they have the dominant allele, they don't have the disease. The third generation, no one's affected. The fourth generation, there are some homozygous recessive offspring, which you know is always what? It's always little a, little a. Okay, so this gives the biologists a general idea or definition of autosomal recessive disorders. And here we go. This is what I want you to know. Autosomal recessive disorders means most children affected by autosomal recessive disorders have parents who are not affected. Okay. Number two, and this is easier than you think. Autosomal recessive disorders require that person have what genotype? Homozygous recessive. Little a, or little s, little s. Two recessive alleles must be present to display the disease. I'll say this again. Autosomal recessive disorders require that these children mostly may have parents who are not affected. Say these children have parents who are not affected. Same here. Not affected. But they're still homozygous recessive. And number two, to have the disease, you have to be homozygous recessive. Okay? All right, so here we go. Examples. Autosomal recessive disorders. Now, again, keep in mind what I just showed you on the pedigree to interpret is extra credit only. I'm not going to give you a pedigree and say, is this dominant, autosomal recessive, or what? Okay. So now, to clarify that definition, autosomal recessive disorders are recessive alleles that must be two, right? So little h, homozygous recessive. And they're required to display, show an autosomal recessive disorder. Now, some of you are probably wondering, well, what does autosomal mean? I'm going to put this into chat because it is equally important. Here we go. Autosomal means that gene is found only on chromosome number 1 through 22. Number 1 through 22. And we're going to talk about this. Don't worry, we'll talk about it. Humans have 23 homologous pairs of chromosomes. So autosomes do not include pair number 23. Don't worry, we're going to go over this a lot. So autosomal disorders are genetic homozygous recessive disorders found on any of chromosomal pair 1 through 22. Example, number 1, Tay-Sachs disease. You don't need to know all this stuff here. You can read about it if you want to. Just know this example. Okay. If you want to study it more, it's actually going to affect neurons and the accumulation of basically waste inside of these oxidized lysosomes. And this affects the function of these neurons. Second example that is also autosomal recessive to know. Number 2, cystic fibrosis. This is probably a little more relevant. More of you might have heard of this. So the first example was Tay-Sachs. Second example, cystic fibrosis. I do want you to know the following about cystic fibrosis. It's the most common lethal genetic disorder among individuals who are white in the United States. Generally of European descent. The reason, extra credit, is right here. Second point or star that I have is extra credit. Cystic fibrosis forms because the chloride ions do not pass through their channels. I do want everyone to know this third point. Cystic fibrosis causes abnormally thick mucus to develop in most of the mucous membranes in the body. So that's including the sweat glands, the digestive tract, the bronchi, the pancreas, and the list goes on. Usually a decreased lifespan because the clogging of these mucous membranes and the tubes get clogged because the mucus is so thick, infections occur, and the infections lead to death. You can look at this if you want to. That is totally optional. So far, Tay-Sachs, cystic fibrosis are autosomal recessive, that is homozygous recessive disorders found on the autosomes. So keyword, autosomes, number 1 through 22. And remember, when I'm saying number 1 through 22 or 1 through 23, I'm talking about pairs of homologous chromosomes. I'm using XSOMES here, shorthand for chromosomes. Number 3, and this is a biggie too. A third example of an autosomal recessive disorder is sickle cell anemia. Sickle cell anemia tends to occur in many, not a lot, but a good number of African American individuals. Okay, sickle cell disease. Here's what you want to know about it. Not only is it autosomal recessive, but the red blood cells are affected. So it's not just any blood cell, it's the red blood cells. The red blood cells tend to look almost kind of sickle shaped, which is like that. So they're irregularly shaped because the hemoglobin is not right. It's not functioning right. And I want to remind you that hemoglobin, and let me put this in chat for you. Hemoglobin is where most oxygen is carried by the red blood cells. Hemoglobin carries most of the oxygen. That's why if we become anemic, that's because usually our hemoglobin is not working or our red blood cells are few in number. So sickle cell disease leads to, this is in chat, leads to anemia and possible shock, blood loss, and a huge need for blood transfusions. These abnormal cells clog blood vessels. The anemia is a big, big sign. They usually, sickle cell patients have to go in the hospital sometimes several times a month. I've had some students who had this and they've lost almost a third of their weight. It's very, very severe. Okay. Constant infections. Here's a picture. All right. So 3 actually very severe diseases, Tay-Sachs, cystic fibrosis, and sickle cell disease to know that are autosomal recessive. Any questions on autosomal recessive disorders? Let's now turn to autosomal dominant disorders. One thing that, and I forget, I think it might have been Justin or Evan answered it the other day in lab about, does having a dominant allele mean that it's necessarily more common? No, 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 it doesn't. It's dominant and recessive, remember, just have to do with how they show themselves. If it's a dominant allele and there's a recessive allele next to it, the dominant allele covers the recessive one up. Recessive only shows itself if there are 2 recessives next to one another. Okay, that's a general rule to keep in mind. Okay, I'm going to put this in chat again. So general rule, remember, dominant allele covers up any recessive allele next to it. But recessive alleles need 2 to display, show their phenotype. Okay. Now, autosomal dominant. Let's get into this. There is a pedigree. I'm not going to go over this, but you will notice something a little different. Some of you who are really observant, in contrast to the previous pedigree back here for autosomal recessive, notice the 2nd and 3rd generation have no disease in anyone for the recessive pedigree. But now autosomal dominant, every generation has the disease. Okay. Every generation, someone, at least one person has the disease, right? That suggests autosomal dominance. Right now, again, I'm not going to ask you about the pedigrees and whether it's autosomal recessive or dominant, but examples. Before we get into this first and second example here, let's define this, okay? This is new, so listen carefully because it's going to go against some of the rules we just learned. I'm putting this into chat. Autosomal dominant diseases or disorders occur when only one when only one allele that is dominant is present. Only one. Okay. Now here's the difference. Then how does that contrast with autosomal recessive? Because remember, autosomal recessive disorders, you need two recessive alleles to show the disease. For autosomal dominant, look at the yellow, shaded yellow places in their genotypes. You need what? You need just one dominant allele. You can be a heterozygote. That means you can be a heterozygote or a homozygous dominant. Sorry about the spelling there. Okay, that's the key. So now that makes sense because a lot of people are heterozygous in their genotype. It makes sense that autosomal dominant disorders will be more common. Okay, so this is a case where actually, yes, they can be more common. All right, so first example, Marfan syndrome. Marfan syndrome. You don't need to know all the reasons and causes behind it. Probably the biggest thing about this is Marfan syndrome. These people tend to have long limbs and fingers. Their skin and connective tissue tends to be able to stretch a little bit more. But probably most significant, they're at risk for very dangerous aneurysms. These aneurysms are like balloons of the blood vessels, and they can burst and a person can die instantly. So Marfan syndrome is considered severe. They have to have regular heart checkups and blood vessel checkups as well. Number two, second example to know. This decreases the lifespan of individuals with this disease, Huntington's disease. Okay. It doesn't show signs or symptoms until about middle age. There's unfortunately no effective treatment for it. It is a neurological disorder. That's what I want you to know about it. And it does decrease the lifespan because the neurological symptoms get worse and worse. Basically, the person, if other people didn't know what they had, they might think they're drunk. But the symptoms of that, the floppiness and, maybe the unsteadiness and the gait get worse and worse, and it keeps going, and eventually their life shortens drastically. So they tend to die around 50, 60 years of age. That's a second example there. And you can see, wow, it's akin to Alzheimer's, kind of like Alzheimer's, but it's different in many ways too. You can see on the right, the neurons in the brain of a person with Huntington's have been lost compared to a normal brain on the left. So two types of disorders so far, autosomal recessive and autosomal dominant. Remember, there are three examples for autosomal recessive, and you just need to know two examples of autosomal dominant, the Huntington's disease and Marfan's syndrome. Now for something that is a little bit different, two actual concepts that are a little bit different. Some of you might have heard of this before. In some cases, they talk about red flowers, pink flowers, and white flowers. Well, here's a good example. This is an example of incomplete dominance. Definition: in incomplete dominance, the heterozygote, so if we're talking about flowers, maybe it's big F, little f, the heterozygote has a phenotype that's in between. In other words, the heterozygote, big F, little f, shows pink in its petals. That's assuming that big F codes for red petals and little f, recessive, codes for white petals. All right, so offspring of a red flower with a white flower, if they have pink flowers, then it illustrates incomplete dominance. Notice right here, the phenotypic ratio is not 3:1. So phenotypic ratio is not the typical 3:1 where we talked about dominant to recessive. Remember, back with complete dominance, it was 3:1. So if this were complete dominance, it would be 3 red flowers for every 1 white flower. But it's not. It's 1 red flower, 2 pink flowers, and 1 white flower chance from the cross of red with white. Okay, so the ratio is 1 to 2 to 1, which means basically 1 out of 4 chance, or 25% chance of having red flowers from the cross above. And when I say the cross above, I'm referring to red parent flower with the white parent flower. Okay. 2 out of 4, which is 50% chance of having pink flowers from that same cross, and your 25% or 1 out of 4 chance of having white flowers from that same cross. So what does a cross look like? The cross basically we could say is FF times FF. And I have this in chat. It's just easier to write it there in chat. Okay, so you can see big F, which is homozygous dominant, represents red flowered parent. And then, of course, the little f is going to be homozygous recessive, which is white. All right. Okay, so big F is homozygous dominant, which is red. Right now, having said that, let's look. There's a beautiful Punnett square. The illustrations always do help. Okay. So you can see that here, this is an example, and it continues to be explained with several more examples. So we've seen flowers. All right, so now let's look at this here, right? And actually let me back up for a second. Let me go back for a second. Let me clarify this just so you can see this. So the initial cross, to be clear, is like our initial cross with the monohybrid cross. So you're going to have an initial parents that cross like this to form what? Big F, little f. That is hybrid, right? Hybrid is what? Pink. Okay, so big F, little f, pink. Parent crosses with pink parent. That is a cross we're talking about of parent flowers that gives you that 1 to 2 to 1 ratio. Now, how did I get these 2 parents who are hybrid? How did I get them? I got them through this cross that I just wrote into chat, which is, and I'm clarifying this for everyone, so notice the chat, homozygous dominant with homozygous recessive, because all the children, all the offspring of the homozygous dominant with homozygous recessive are going to be heterozygous, right? Which is big F, little f. So red with white, red flowers with white flowers crossed that are purebred. I just did it. The big F with little f gives pink flowers. Pink flowers are big F, little heterozygous. That is what we see as the parents right here. And now the only thing you need to understand is that since this is incomplete dominance and not complete dominance, the ratio is 1 to 2 to 1. Okay, there's your 1. Here are your 2 pinks and your 1 white flower. So 1 red, 2 pinks, and 1 white flower. That's the result of crossing 2 hybrids. So in other words, the basic gist is the hybrids, the heterozygotes, show a phenotype in between dominant and recessive. I'll say that one more time. It's so important. Hybrids, which are heterozygotes, show a phenotype in between the dominant and recessive. In this case, it's in between red and white. Therefore, it must be a phenotype that's pink. That's incomplete. Now, a few more. Multiple allele and codominance. These refer to blood types. Now, remember, we've already talked a little bit about blood types, okay, in lab especially. And I want to remind you that there are 4 blood groups, but the 4 blood groups are not the same as the number of alleles. Okay, so remember we talked about antigens. So I'm adding this to chat. Number of alleles, blood cell alleles. There's going to be your A, right? Your B, and something else. This is new on this new slide. We could call it O, but we're not. We're going to just call it I. I is the 3rd allele. Now, this particular slide is, I have number 44 on this. This lists the ABO antigens or alleles. These are not the blood types. What I have with the rectangle, the vertical rectangle, are not the blood types. Blood types are right here where the arrow is. Okay, these are the types. And you already know there are 4 blood types. And you can count them right there. If there are 4 blood types, how did they get those 4 blood types? From 3 alleles. Okay. So let's list those alleles. They are IA for the A antigen, IB for the B antigen, and little I, a 3rd allele for neither A nor B. Okay. Now I'm not going to ask you these, I promise, because this is pretty detailed. But if you go into general biology, you do have to understand this. The point I'm trying to make is this. In order to get the 4 types, the 4 blood groups or 4 blood types, they come from a mix, especially A and B, of genotypes. So for example, notice if you're type A, you can either be homozygous dominant or heterozygous. What's that? That's having 1 dominant IA allele and 1 little I allele, but still you are blood type A. I know it seems strange, but different genotypes for A, different genotypes for B. Yes, Jose, blood type and blood group are the same. Okay. So the three listed here, though, be careful. Those three are alleles. So the ones that say IA, IB, and little I, they make up the four blood groups, blood types. Okay. Look at B. Notice two different genotypes. Homozygous dominant, 2 IBs. Heterozygous, 1 IB, and a little I. It's interesting because if you're AB or O, you only have one genotype. You don't have two. So 2 genotypes for A, 2 possible genotypes for B. That might be extra credit. Inheritance here, I'm not going to go through that. You can look at the Punnett square, but in general biology, they are required to understand that. But it does explain, if some of you ever wonder what your parents' blood type might be, you can actually figure it out from some Punnett squares and doing that if you wanted to. You can look at it. I'm not going to ask you that on any test, though. So multiple alleles refers to the fact that we've got 1, 2, 3 more than 2 alleles coding for blood groups. Second term, codominance. So codominance is when the alleles are equally expressed in a heterozygote. And we saw this right back here. Let's look at blood type AB. In other words, IA equals IB. That's what codominance means. I'll say that again. It's so important. Yes, these are multiple alleles, which express codominance only between IA and the IB allele. One more time. I'm going to make sure you all get this. These are 3 multiple alleles, but there's codominance only between the IA allele and the IB allele. Codominance, which means both alleles are equally and fully expressed. Equally and fully expressed in a heterozygote. So blood type AB only expresses each allele fully because their blood cells contain the A and the B antigen. So both products are fully present. A and B are found on all red blood cells. Codominance. All right, we're almost done. Polygenic inheritance. So you can see this can get pretty complicated. Yes, it can. Poly means many. Genes mean many genes. So many genes influence inheritance. Definition: when a trait is controlled by at least 2 or more genes. And remember, each gene usually contains at least 2 alleles, like big A, little a. So 2 or more sets of alleles. These dominant alleles are going to have a quantitative or additive effect. The result you want to know is a continuous variation or really stream here of phenotypes that we'll see. 2 examples to know: height and skin color. They are visible on a spectrum. So here they happen to be showing skin color. And you can see the variation in spectrum, but it's a gradation. It's a continuum that is quantitative or additive. And yes, there's an area where it's kind of like a single unimodal curve. There is an area where there's most individuals, most organisms have something that's somewhere in the middle. And then fewer and fewer organisms have the more extreme variety of those skin tones. And you can see this is all based on the combination of these alleles. So polygenic inheritance, a number of pairs of genes is influencing this. In this case, it's the A gene, the B gene. Notice I don't write two alleles for these because it could be either or. And the C gene, according to this. Again, it could be big C, big B, little b, big A, little a. So it's a bell-shaped curve. This is only considering 3 genes, but in reality, it's probably 20 or more genes that influence this. That's not even counting environmental influences. So let's start ending this with environmental factors. So environmental factors are going to influence phenotype. So phenotype is also influenced by the environment. That is outside, non-DNA factors. So nutrition is one, temperature is another. And even distinct from that is sunlight. Sunlight, humidity, the list goes on. Polygenic traits like height and weight, they're influenced by these environmental factors. So one thing we know for sure that, yes, over time, over the last 2 centuries, human height has increased in general on average, partly because of nutrition. Certain flowers and their color is influenced by temperature. And guess what? You all saw that in lab with those bacteria on the plates. Some of them were kind of whitish gray. Others were red. That was due to temperature influencing genes. In other words, temperature, for example, temperature can activate genes or turn off genes. That's the beauty of it. And here's a good one for height, as you can see. And they're just using a small class here, a small class sample, in order for the curve to look more like what you see here. You need about 1,000 more people. The more people you have in your sample, the smoother that curve will be. It won't look really dilapidated like this, you know. It won't look all strange like that and bumpy. Okay. All right, we're done with that chapter. Before you go, I want to show you all something very important for chapter 24, because we're getting into that next. We're going to briefly in chapter 24, which is Monday, talk about linkage. I do want you to review over the weekend homologous chromosomes. So I'm putting this into chat. Homologous chromosomes, we're going to be jumping right back into them again and talking about karyotypes on Monday. Homologous chromosomes, remember, are the identical maternal and paternal chromosomes found in our DNA. Each maternal and paternal homolog, you see one here, must carry identical genes, but not necessarily identical alleles. Okay. So what you're seeing right here could be the paternal, it could be the maternal homolog of another chromosome that should be pretty much the same size and the same width and have the exact same genes. These are the gene names found along chromosome 19. And these really are, these are real genes that they've discovered. So just a heads up, we're going to be talking about sex linkage. And then we're really going to get into a karyotype. I'll give you a brief definition now before we go, but the karyotype is a display of all 23 chromosomes, so 1 all the way through pair 23 chromosomes. And notice these are pairs. You'll also notice that they have similar lengths. They stain similarly because they have the same genes. And they get shorter and shorter as you get to the higher numbers. So pair 1 through 22 are the autosomes. Okay. Pair number 23 is the sex chromosomal pair. That is either XX or XY. And that's this right here. So this is from a genetic male, XY. We'll talk more about that on Monday. And this is really cool because when we talk next Monday and Wednesday about these, we go into a lot of other disorders. For example, Down syndrome and chromosomal disorders. So it's really neat stuff. All right, I will let you all go a little bit early today, and there will be a recording I'll post over the weekend for you. Have a good weekend. Monday, we're going to be looking at chromosomes in a sort of making your daughter game in lab. Don't forget the homework as well for lab about the questions. Remember about the tongue rolling and the finger, digital hair, all that stuff. 5 to 10 people is totally fine. All right, have a good one, folks. Any questions? I think Thomas, you had a question. Feel free to, you can unmute if you want to. I think, Thomas, you had a question at the very beginning or something.
Hey, Ms. O, can you hear me?
Yes. Hi.
Okay. I don't know how to use this stuff, so I was trying to
Oh, don't worry, I don't either.
at the beginning of class. But I had a question, Ms. O, for the codominance.
The what?
The codominance.
Yes. Codominance.
So that means like they share or live together.
Now, codominance is this, is type AB blood.
Okay. Yes, I have that written down.
Because what do you have it written down as, Jose?
I put 3 multiple alleles only IA and IB codominance?
Exactly, because type AB blood, and I'm putting this into chat,
Okay.
type AB blood has IA, IB.
And then I put the both alleles is equal and fully expressed in the hetero, but what the fully expressed, does that mean like they show up,
They
or
show up. Yes, you got it.
okay,
Right.
and they need the heterozygote to be able to do that?
Yes, the type AB. Yeah,
Okay.
you got it. I can show you again Monday because you don't have the test for another week or two, but I'll show you again on the board Monday.
Mhm.
Okay, remind me, please.
Okay, so I might have to, if it's okay with you, I might have to go over uh Mondays and today's, because I'm having a hard time uh
That's okay.
trying to
I'll
get this.
go. No worries. I'll go over it for
Okay.
sure.
I already had a hard time with the blood, so
It's tough. Genetics is not easy. I know. And I'm really simplifying it compared to regular biology. So
thank you.
yeah, yeah.
Okay,
Be
Ms. O.
sure to ask me Monday.
I will. I'll maybe after lab, or a class or something, or after lab, uh, probably go over or something. But thank
That
you, Ms.
sounds
O.
good. That sounds good. I'd be happy to explain it.
Okay, thank you. Have a good day.
You're welcome. Have a good week.
You too.
Take care.
Bye.